So we've talked about the steps in phagocytosis. Now we're going to talk about how microbes evade this process. They can basically do it at any point along it, depending on the organism. They have that ability to overcome. So first, we have the ability to inhibit adherence, aka the phagocytotic cell can't grab a hold of the bacteria cell. That would happen if they had an M protein or capsules. They can also kill the phagocyte by releasing a chemical. They can lyse the phagocyte. In this case, they're going to blow holes in the membrane. They're going to use the membrane attack complex to cause this. They can be brought into the cell and then actually escape the container, the phagosome. This basically allowed them a door into the cell in the first place. They can stop the phagosome and lysosome from fusing, aka stop the addition of the lytic enzymes into that little container. They can survive in the phagolysosome. In this case, they actually are able to overcome those chemicals, those digestive chemicals. So here's a video looking at hiding from host defenses, at getting past that phagocytotic process. So we're going to zoom in into the body. Here we have our phagocyte. Here we have that foreign particle. Here, in this case, it looks like a bacteria cell. They're going to grab a hold of it, bring it into the cell, put it into a container, the phagosome. That container is going to have a lysosome fused to it, which is going to add lytic enzymes. That's going to break apart that cell. The pieces are going to be spit out of the cell if they're unusable. Now, we also have some other processes where B cells and T cells identify bad guys. And some of the things that we've mentioned would allow them to overcome this process too. And we're going to talk about B cells and T cells a little bit later, so I'm going to skip that part. Here we have the example of a capsule. So you can see the bacteria cells moving around. The phagocytotic cells would come into contact with it. If that cell has a extra organized glycocalyx or a capsule, then that's going to make it more difficult for them to grab a hold of it. So here we have the phagocytotic cell trying to grab it. It can't. And here's one that doesn't have a capsule and it grabs a hold of it, brings it in, it goes through the process. Now they can survive phagocytosis, so bring them into the cell so they've actually been taken up. Once inside, they can stop the lysosome from binding. They can get out of the phagosome. They can let the lysosome bind, but the enzymes don't have an effect. They just get to populate that container inside the cell. Now we mentioned that the organisms are able to sometimes change their name. We talked about an antigenic variation before. So here's a picture of the surface antigens. So right here on the outside, those are markers, those are names of those cells. And we have some of our immune cells hanging around, including antibodies. If we have those immune cells, they're going to recognize a particular part of that invading particle. In this case, we have an antibody. It doesn't recognize the first virus, but it does the second virus, and that's going to mark that virus. Here we have trypanosoma, and we have a glycoprotein, or a name, an antigen on the outside. And if you'll notice, there are a couple with the same name, and one with a different name, a different marker, a different antigen. If we're producing cells that recognize this one, it's going to recognize everyone with that marking, that antigen on the outside, but it won't recognize this one. So this one will be able to populate, and then when we start recognizing this one, there'll probably be a different one produced. So recognize and destroy, but not that one. It produces offspring, and then those offspring can change their antigens and be able to survive the next wave of assault. Inflammation is another way that our body fights infections. And inflammation is caused by acute phase proteins that are activated and then cause a cascade effect, and we get the outcome of this process. So we get vasodilation. Vasodilation is where our blood vessels actually become leaky. This allows the things that travel through our system to actually get out into the areas that they need to go. Now we have signs and symptoms of inflammation: redness, swelling, pain, and heat. What this does is we recruit the molecules, the cells, that are actually going to fight foreign invaders to the location of the inflammation. That swelling is where we have that leakiness occurring. We have those molecules being able to get out of the highway of our bodies into that tissue. This allows us to destroy whatever it is that is foreign and limit its effects on the body by maintaining it in that area. It also starts the process of repair to replace the tissue damaged by whatever it was, whether it was a cut or directly by a microbe damage. These chemicals are released by damaged cells, and they cause the processes that lead to inflammation, whether it be vasodilation and that increased permeability of the blood vessels, allowing things to move out into the area. It can be other chemicals that actually intensify their effect and encourage phagocytotic attachment. So here's just a picture showing the process of inflammation. We have a cut, and in that cut we have the possibility of bacteria. Now, that is in the tissue, so in the tissue here. We also have our blood vessels, that's the highway throughout our body. We have some nerves. We have our epidermis, our outer layer, our dermis, our inner layer, and then our subcutaneous tissue there. Now, the chemicals are going to be released, and they're going to be released into that area of inflammation. That's going to cause a swelling to occur here. By releasing them there, they're going to call in the reinforcements, and they're released by the damaged cells. We end up with a blood clot and a abscess. Vasodilation is going to allow the traveling of other molecules to get to this area, traveling of cells and other response units. Here's showing that vasodilation. So literally we have this structure, this container, this blood vessel that things are going to be moving through. And that includes our monocytes, our macrophages, our neutrophils. They're going to travel through this system. And what it does is it allows them to get anywhere that this blood vessel would come into contact with. Vasodilation opens up the barriers, the cells that outline the outside of the vessel. Open those up so those cells that need to respond can actually squeeze through and get out. So we have margination, where the phagocytes stick to that outside of that vessel. Then the phagocytes actually squeeze between those cells, and they hopefully go and eat the bacteria that's out there. Here's a video showing the inflammation steps. So here we have a damage to the tissue and the insertion of bacteria. Those bacteria are going to multiply and divide. We're going to have chemicals released. Those chemicals are going to come into contact with the blood vessels, where it's going to cause vasodilation, the swelling, and the opening between the cells. The phagocytes, blood, and other things will be able to get out of the vessels and go to the site of damage. Once they're there, they're going to be able to engulf and destroy, hopefully, any foreign invaders. Pus will develop and will clog the hole, and then the tissue will regenerate. So here's the tissue repair. We have a scab and we have a blood clot. We're going to regenerate the epidermis. Now, fever is just an abnormally high body temperature. Normally our body is set at 37 degrees Celsius. Phagocytes, in reaction to a gram-negative endotoxin, will actually release interleukin-1. So here's just an example. The hypothalamus releases prostaglandins that reset the hypothalamus to a high temperature. The body increases the rate of metabolism. That's going to, well, allow us to get more energy. We need that to be able to generate the high temperature and shivering. Vasodilation and sweating are actually used to cool down our body temperature or go through crisis after we've raised it high. We can't handle our body temperature staying high for very long, so we have to have a way to fix it. So the advantages of fever is it's going to increase chemical signals, produces the interferons, but the disadvantages are going to be our heart not being able to handle it, a change in our pH, dehydration, and of course the higher temperatures are going to damage our actual proteins, damage our cells overall. Now the complement system is one of these messengers, one of these ways that we can actually cause things to happen in this process. So these are some serum proteins, and they're activated in a couple different ways. But once they get activated, we will have a cascade event where one molecule activates more molecules, which causes an outcome, activates more, and causes a final outcome. All right, so here we have a visualization of complement and the process. We're going to start out with the classical pathway, the basic way that it gets started. Now we have a foreign invader here. It has antigens on the outside. Those antigens are going to be recognized by antibodies. The antibodies are basically targeting that and saying it shouldn't be here. Now in this process, C1 gets activated by these antibodies, and C1 will cleave C2 and C4. C2A and C4B bind together to actually produce the next molecule that does a job. It's going to then interact with C3, which it breaks into C3A and B. C3A and B can actually do jobs, but first let's say what else they do. And that is that they cleave C5 into its part A and B. Now C5B binds to C6, C7, and creates a final outcome. Now we're going to talk about exactly what these guys do in a moment. In the classical pathway, this process begins with the antibodies. So we have all these C's floating around the complement molecules. We have a bacterium. We're going to recognize that bacterium with an antibody. Binding to antigens, C1 gets activated, breaks C2, C4, gets C2A, C4B, breaks C3, C3 breaks C5, and then we get the final C6, C7. Now in the alternative pathway, something else activates. Now in the book they talk about these B, P, and D. There's more to it than that. It literally happens that something causes C3 to get activated. So notice it skipped the first steps. So C3 gets activated, and once C3 gets activated, it's going to cause the rest. C3 activates C5, and C5 does its job, C3A and C5A does its job, and C3B does its job. The same outcome occurs. Now what's interesting about this is C3 gets activated periodically, almost by accident. And it's just one of those things to make sure that there's nothing we haven't identified yet going on. Our own cells can't be damaged by this. We have a way of actually disassembling this process if it were to target one of our own cells. Now in the lectin pathway, we have carbohydrates on the outside of the cells that's bound by lectins. Those lectins are going to activate C2 and C4. They're going to go together just like they did before and activate C3, and then we get the activation of C5. So C3b causes opsonization, which allows us to mark the pathogen. C3b literally binds to the invader and marks it as something that can be grabbed a hold of. It makes it easier for the phagocytotic cells to grab the pathogen. C3a and C5a go along on their merry way and actually help cause inflammation. They're going to cause chemotaxis and the bringing of all the immune system molecules to this location. C5b binds to C6, C7, and C8. Together, they recruit a ton of C9, and it's called the MAC complex, and it's in this picture down here. So if you'll see, here's that C5 through C8 bound there, and then here's a bunch of C9s. And basically those C9s, a bunch of them join together and make a pore or a hole in the invader's phospholipid bilayer. This will allow the contents of the inside of the cell to come out and will cause damage to that cell. So cytolysis is the outcome of the C5b through the rest of the complement. So here's a visual that's going to allow us to show the results. Again, opsonization, marking the bad guys. Cytolysis or cytolysis, which is going to be damaging, poking holes in the membrane of the foreign invaders. Chemotaxis and inflammation, bring in the rest of the cavalry. So we have our process, everybody's getting broken, C3b is binding, etc. Complement is being activated. Now first, chemotaxis and inflammation. C3a and C5a are going to help bring in the phagocytes and cause inflammation. Recruit the cavalry. Opsonization is where we mark a cell, which makes it easier for the phagocytotic cells to grab a hold of it. Cytolysis or cytolysis is where C5b, C6, C7, and C8 bind to the cell and recruit a ton of C9. That's going to allow it to poke a hole through the plasma membrane, through the phospholipid bilayer, and the contents of that cell will come out. So here's the damage caused by it. The outcome, again, I've already said, opsonization or immune adherence means that we get enhanced phagocytosis. Cytolysis or cytolysis caused by the membrane attack complex is going to damage the cell. Chemotaxis is going to attract the phagocytes, and then of course inflammation is going to allow those phagocytes to get out of the vessels. Just a summary slide showing exactly what I said before with those pictures and the outcome. I do expect you to be able to tell me that C5b through C9 are the ones that cause damage to the cells. They make the MAC complex, membrane attack complex. That C3b is the one that marks microbes, and that C3a and C5a are going to play a role in inflammation and chemotaxis. Here is how C5a and C3a bind to histamine-releasing mast cells and actually cause them to release histamine. We also have C5a, again, recruiting macrophages and neutrophils or phagocytotic cells to come into that location. Some bacteria can evade complement. Their capsules prevent the actual activation of the system. They can prevent the membrane attack complex from forming, and they can actually break down C5a. We mentioned interferons. Interferons are important for antiviral protection. So what happens is if we have a cell, so this cell right here has actually been invaded by viruses. And what happens is that cell will send out a message saying, I've been taken over. Protect yourself, lock down all your windows, close all your doors, and produce antiviral proteins and stop from being infected. IFN gamma, or interferon gamma, causes neutrophils and macrophages to phagocytize bacteria. So interferons are going to cause different reactions depending on which interferon we're talking about. We also have iron binding proteins. So I mentioned before, we mentioned this before, that iron is important. And if we are able, and what we do, is we bind iron. There's not very much free iron in our system. So in order for the microorganisms to be able to survive, they're going to have to be able to do something or steal the iron. So transferins actually bind serum iron. We find them in the blood and tissue fluids. Lactoferrin is found in milk, saliva, and mucus. Ferritin is found in the liver, spleen, and red bone marrow, and then hemoglobin is located in red blood cells. All of these are getting iron bound up so it's not available. What happens is bacteria produce centrophores that can actually steal the iron from these proteins. They can also digest red blood cells. Now we also produce antimicrobial peptides. We will find these in our saliva, in other places in our bodies. These are short peptides that are produced in response to protein and sugar molecules on microbes. They inhibit cell wall synthesis, and they make pores in the plasma membrane, which is going to lead to cytolysis or cytolysis. They also tend to be broad spectrum. They're not specific for one type of microbe or another, but they're going to recognize general proteins and sugars on the microbes. We first began with a description of innate and adaptive so that we had some background. And then we talked about how the first and second line of defenses work and antimicrobial substances. Thank you very much for paying attention, and good luck.